We now start the panel talks, 4 short talks from Stanford Medicine. The first speaker is Hiro Nakauchi. Hiro is a pioneer and a leader in the field of hematopoietic stem cells, and as you will hear, also tissue stem cells, including liver. He was the director of the Stem Cell and Regenerative Medicine Institute at Tokyo University, and we have been very fortunate to have him back at Stanford since a year. So he's now a member and a professor at Stanford Medicine and the Institute for Stem Cell Biology and Regenerative Medicine. Hiro, thank you for being here.
So I start with some background slides. As you know, hematopoietic stem cells reside in a very special bone marrow microenvironment called stem cell niche. And their fate, such as self-renewal, differentiation, hibernation, or proliferation, are regulated by this special environment called hematopoietic stem cell niche. So it's very important to understand what are the components of this niche and what is the mechanism to regulate HSCs. So we've been interested in this hematopoietic stem niche for a number of years. And several years ago, we encountered a very interesting paper published in Science in 1946, so it's almost 70 years ago. The first author was Arthur Kornberg, our professor emeritus and Nobel Prize laureate. And what he found in this paper was that when the rats fed with low protein diet, they developed very severe granulocytopenia or anemia. And when he gives a mixture of purified amino acid, that corrected this disease. So the first demonstration of relationship between amino acids and hematopoiesis. And since then, no paper has been published. And luckily to us, Arthur Kornberg didn't go any further. So I think he was more interested in folic acid and DNA synthesis, which led to his Nobel Prize later years. So we decided to study on this amino acids as one of environmental niche factors crucial for the function of HSCs. The first experiment we did is an in vitro experiment. We cultured highly enriched stem cells with complete media containing over 20 amino acids, and also 20 different media, each of which depleted one amino acid, so single amino acid deficient media, 20 of them, and also the medium without any amino acids. After 1 week of culture in the presence of SCFTPO and the serum-free condition, we counted the cell numbers in each well. So this is the result, and which was quite surprising to us. Actually, in 2 wells, we did see severely retarded cell growth. One with depletion of amino acid X, and the other was amino acid Y. Because these are all new and unpublished data, I'm not able to disclose what they are at this moment. Sorry about that. But anyway, this is quite surprising. So when we went on to see its effect in vivo, so I put C57 black 6 mice on either amino acid X depleted or amino acid Y depleted diet for 4 weeks, and we examined their bone marrow cells for the frequency of hematopoietic stem cells. And as you can see here, we didn't see much changes with the diet depleted with amino acid Y. But with amino acid X, there was significant decrease in the hematopoietic stem cell number. And on the right-hand side, you can see almost as early as 1 week after initiation of this special diet, we did see a decline in HSC numbers. This slide shows the bone marrow morphology, and only the mouse bone marrow from the X-depleted diet showed a significantly decreased cellularity in bone marrow. The influence of this depletion of X is also found in lymphoid organs. For example, in the spleen, in the right-hand corner, we could observe destruction of the lymphoid follicle structure, and spleen is very small, and thymus was almost undetectable. So it is very significant in vivo effect as well, just 4 weeks of depletion of amino acid X from the diet. To further validate these findings, we performed competitive repopulation assay. So here we put B6 mice on X-depleted diet for 3 weeks, and then we transplanted a whole bone marrow cell into the lethally irradiated congenic mice, along with the control, equal number of control whole bone marrow cells. And 3 months later, we use these genetic markers to discriminate competitor and donor cells to calculate the contribution of these cells. And this is the results. Again, up to 2 months post-transplant, both populations, both bone marrow cells contributed almost equally for the reconstitution. But at 3 months post-transplant, we could see a very sharp, dramatic decrease in their contribution. So these data indicate that influence of this X-depletion diet is rather restricted to long-term HSCs, but not so much on progenitor population. This is, I think, a very important observation. So I have to explain that HSC niche is also critical for successful hematopoietic stem cell transplantation. As you know, we have to do myeloablative pretreatment to vacate, to empty the HSC niche. But this myeloablative treatment, such as high-dose irradiation or high-dose chemotherapy, is a major complication of this treatment, and this limits the indication of this therapy. So we've been looking for a way to avoid this myeloablative preconditioning regimen. So, if you think about the fact that dietary depletion of amino acid X reduces the number of HSCs, we thought that it might, be able to replace the myeloablation in the setting of the hematopoietic stem cell transplantation. So we designed a mouse model of allogenic HSCT to immunodeficient patients, because they are the patient who really needs a non-ablative HSCT. So we put NOD skid mice, they are the immunodeficient mice, on X-depleted diet only for 2 weeks, and then we transplanted allogenic stem progenitor cells into this NOD skid mice. And then we followed them after switching to the complete diet. As expected, we could see long-term engraftment by the donor cells. As you can see, essentially 100% of survived recipients showed engraftment with relatively high donor chimerism, whereas the control mice recipients didn't show any engraftment as expected. And I'm not showing the data, but all the lymphoid organs are also repopulated very nicely after the transplantation, but without any myeloablation. So lastly, and most importantly, we tried to see if the same amino acid X influence on the human HSCs. So first we prepared humanized mice by injecting cord blood CD34 positive cells into the NSG immunodeficient mice. After confirming the engraftment, we put them on 2 weeks of X-depleted diet, and then we analyzed peripheral blood mononuclear cells. And on your left-hand side, the FACS plot shows that actually, there was not much difference in the human to mouse ratio proportionally. But on the left-hand side, we observed a significant reduction in white blood cell numbers. So we were able to recapitulate what the observation made by Arthur Comberg almost 70 years ago, just by depleting single amino acid. So in conclusion, amino acid X and Y are critically important for proliferation survival of HSCs. and dietary restriction of amino acid X alone enabled non-myeloablative HSCT in a mouse model. And human stem progenitor cells are also dependent on amino acid X for proliferation and survival. So the future direction is to elucidate the mechanism of amino acid X dependency unique to HSCs, and also search for similar amino acid dependency in leukemic or solid tumor stem cells. So the work I presented today is done mostly in my lab in Tokyo by the two students of mine, Yuki Taya and Satoshi Yamadaki. So I stop here. Thank you for your attention.